The most abundant element in our atmosphere just so happens to be one incredibly crucial for the survival of plants, and by that way, all the way up the chain, all life. If you haven't already known this, it's nitrogen, an element that was a serious constraint on agriculture pretty much time immemorial until a new method came to extract it out of the air in 1913. Most plants rely on nitrogen to be available in their soil, while others still, like the beautiful redbud tree, have little organisms within them to help convert the nitrogen into a form they, and by way the surrounding plants, can use. These plants are in a special class called nitrogen fixers, as they fix the soil with nitrogen. Extracting nitrogen out of the air requires industrial processes and equipment, and energy to run them. While the gaining method of regenerative agriculture that I've talked about several times on this show, and thankfully is gaining momentum, smartly balances nitrogen fixers with plants that require it in order to have a homeostasis with the environment. Instead of having to add chemical nitrogen, which can have disastrous effects like algae blooms, just look at the runoff of the Mississippi River into the Gulf of Mexico. This would have none of those, and can go on and go on and go on. Think about planting, for example, one redbud tree for every 3 apple trees. That would create an environmental homeostasis in which those apple trees are getting the nitrogen that they need and then able to produce the fruits that we enjoy. When it comes to the ocean, mind you, most life is actually confined relatively near the coast. This is partly because nitrogen limiting the plants that can grow there. Of course, there's other factors. But it's easy to cut and say in a group, nitrogen is definitely one of the factors. A recent discovery added a new way of getting nitrogen into the mix by these creatures called nitroplasts, or a little bacteria that within their body lives another organism. And that little organism acts as an engine to create all the nitrogen that both of them need in exchange for energy to survive. Put another way, these nitroplasts have a creature living inside them, where in exchange for doing all the work to give them energy to stay alive, they generate all the nitrogen they need. A wild makeup. Connecting it back to us, it's actually similar to how mitochondria work within all of our cells and all other organisms. A process that gave way to nearly all plant, animal, and fungi life, wherein we get energy from the mitochondria and we give it everything it needs to do that. What's fascinating and wild about the discovery of nitroplasts is actually how ancient these organisms are. This wasn't just something that cropped up, but something hiding in front of us in plain sight for as long as Homo sapiens have walked the earth, and longer still, going back to deposits of them, even found on the tops of mountains. My guest for this episode is Tyler Coale, a postdoctoral scholar at UC Santa Cruz, and one of the researchers that worked on the discovery of nitroplasts. In this episode, we get into what they are, how they were found, where they live, and just what they do. This is just the start of the research, and surely will be a wellspring of fascinating information going forward. Thank you, Tyler, for coming on the show. Thank you all for listening. Wherever this finds you on our big, blue, beautiful planet, I hope you're doing well. Take care. All right, I gotta do that cliche thing before the episode starts. If you like what you're hearing, please follow the show on whatever platform you're hearing it through. And if you would, give us a rating as well, as both of those help keep the show going and get it found. One last thing, if you want to submit a topic for me to cover, throw me a person you want me to interview or talk with, even if you want to talk with me, or you just want to tell me how terrible of a job I did on something, check out bandwidth.productions to visit the site and drop us a note. All right, cool. Well, thank you again for taking the time. If you wouldn't mind, could you just introduce yourself really quick?
Yeah, sure. My name is Tyler Coale, and I'm a postdoctoral scholar at UC Santa Cruz in the lab of John Zare.
Nice. I haven't been out to Santa Cruz, but I hear it is gorgeous.
It's a lovely place to work. Yeah.
Well, okay, so I want to talk to you about nitroplasts. But before, I ask everybody when they're on the first time the same question. So I'll ask it to you, which is, what do you like to do that makes you happy?
Oh, that's a great question. I'm a tinkerer. So besides being a scientist and working, in a lab studying microorganisms, I love building things out of wood and metal, and spending time in the natural world. And I think both those things are great kind of distractions, from my work, but they also give me time to think about my work, which, a lot of this type of work, being a scientist, I think a lot of the work happens when you're not actively in the lab. It's thinking about it on your downtime, when you're doing something else. So yeah, I like to distract myself building things and being outside.
Oh, that sounds great. No, I mean, it kind of reminds me of, I think about this a lot, Henry David Thoreau's essay on walking and the importance of
Mm.
doing that. And, it's also interesting. A lot of the most prolific scientists, inventors, engineers, they all kind of did the same thing. They'd work intensely on something. Like I've been thinking
Mhm.
a lot about Einstein and how Einstein would be working on something and then he'd move over to a piano and just start playing some jazz inspiration and come back and be like,
Uh-huh.
oh, I got something, and then go
Yep.
back.
Yeah, I mean, the brain works in mysterious ways, and it's a hard thing to force. You kind
Yeah.
of need to give it space to work on its own terms, and
Yeah,
break things up.
definitely got to let it breathe. And I think
Mhm.
one of the things I kind of rage against, which is actually what I was thinking about with that Einstein thing, is we expect so much of our world to be very reducible and repeatable when
Mm.
in reality we're humans, and I feel like we're expected too much to act like machines. And the reality of being humans is we got to take some time to go for a walk or build something or just put our
Mhm.
feet in the ground or any number of things to disconnect.
Yeah, yeah, I agree. I like that. We're not machines. We shouldn't try to be. We can let machines be machines.
That literally was the vein I was on when I was having that thought. Okay, so I want to get into nitroplasts,
Mhm.
and I found out about these honestly. I think like 2 or 3 days before I emailed you, and I was absolutely blown away by it. Mostly, I mean, my way of doing it is I find mitochondria really interesting, and
Mhm.
so I want to pose to you what I think my understanding of it is, and then to get us started, and maybe you can just correct all of my assumptions and where I'm wrong.
Yeah. Okay.
Okay, so my understanding is that a nitrogen fixing bacteria, so a bacteria that has the ability to extract nitrogen from the environment, has merged in some way with algae to become, and this is the part where it starts getting hazy, are they a new species? Or regardless, they have a new symbiotic relationship. And that process, as I understand, is called endosymbiosis, and that it's
Yeah.
incredibly rare, but has led to some
Mhm.
giant leaps in evolution, like mitochondria, and which mitochondria gave rise to life, animal life, as we know it.
Yeah.
Where in there am I wrong, and what color would you be able to add to help make sure we have
Yeah.
a
I mean,
basis?
that's a great place to start. Yeah. Um, yeah. So you're exactly right that the you know the development, the evolution of the mitochondria really revolutionized life on Earth, and it did give us all animal life, but it actually gave us much more than that. So it evolved eukaryotic life, which is a term that describes cells which are more complicated than bacterial cells, um, and they have essentially more structure inside of them, more organization, and these compartments that allow um, specialized biochemical reactions to occur. That's kind of the innovation of the mitochondria, was a compartment that specialized to form aerobic respiration inside of a cell that then allows the rest of the cell to perform other biochemical reactions um, and really allow it to become more efficient, more complex. And so, really, what the mitochondria gave us is not just plants, well, not just animals. It gave us the lineage that evolved into animals, into plants, and really anything that's more complicated these simple bacterial cells. So it was.
Oh, wow.
It actually happened way before anything we might really call an animal. That typically refers to things which are multicellular. This was an innovation that occurred in single-celled organisms far before multicellularity evolved. So it's even more fundamental than that. But you're right. When you mentioned this is a very rare thing, it happened with the mitochondria. It happened later with the chloroplasts, which those are another instance of this phenomenon that you correctly call endosymbiosis, but it's more specifically it's primary endosymbiosis, which we could get into those details, but it is the it's kind of like you say, it's the merger of two different organisms into one. And so the chloroplasts are this other example of that, and they brought the capability to perform photosynthesis to eukaryotic cells, which eventually gave us the plant lineages and many different algal lineages. And so those two, the chloroplast and the mitochondria, are the classic examples of primary endosymbiosis. The nitroplast is a new example of that, and as you mentioned, it is a different type of chemistry that this example of primary endosymbiosis brings to eukaryotic cells, and that is the chemistry of nitrogen fixation. And I'm kind of if you remind me, if you had other points you made there, I can't quite remember.
No, I mean that was the basis, just kind of giving us a footing of it. But to take it the next step further, so what is nitrogen fixing, and why is it so important? Like, why would algae want to take that ability on?
Yeah, that's a great question, and this is why we, it's kind of the impetus for this research in the first place, is the significance of nitrogen fixation, especially in the marine environment. So this lab, the lab I work in with John Zare, has been studying this process in the oceans for decades, and the importance of nitrogen fixation in the oceans comes down to the fact that there's very little bioavailable nitrogen in the oceans. So this is the reason why large regions of the oceans are, that electric, clear blue color. It's because there's not a lot of plant life growing there. There's not a lot of algae or phytoplankton that's able to grow in the oceans, largely because there's not enough nitrogen, specifically bioavailable nitrogen that organisms can use to build their proteins, build their amino acids, and that's required for their cells to function. So most of the oceans, especially the regions far from land, are very clear. They're very blue because there's not a lot of nitrogen there. But it turns out that certain microbes have the ability to take nitrogen from the atmosphere, where it's very plentiful. The atmosphere is a huge reservoir of nitrogen. And certain organisms can capitalize on that. But to do so, they have to overcome a really kind of significant hurdle, which is breaking apart that nitrogen gas into a more usable form. So nitrogen gas in the atmosphere, N2, is two nitrogen atoms which are connected by a triple bond, which is a rare type of bond that's very difficult to break. And certain organisms have figured out how to break it using a molecular machine, a protein called nitrogenase, which is it's a very complicated enzyme complex. It's got a bunch of different parts to it, but it is able to break that triple bond, convert nitrogen into ammonia, which is a plant fertilizer. And Therefore, organisms that can do this can escape nitrogen limitation in large regions of the world's oceans where there's not this nitrogen that they can just take up from the environment. So it gives them a competitive advantage, and it can kind of form the basis of oceanic food webs. These are the organisms at the bottom of the food chain making their own fertilizer, which then can turn into biomass that fuels the food web. So that's what got our lab and my advisor John Zaire interested in nitrogen fixation many decades ago. And so for decades, this lab and others have been surveying the ocean, looking for organisms which perform nitrogen fixation. And that's kind of the beginning of this research.
That's fascinating. Yeah, I occasionally I'll go really deep in a subject, and recently it was sperm whales, of all things, that I've been getting really interested in. And from that, I didn't realize just how much of the ocean is actually, I wouldn't say empty, but it's a lot more empty than I was expecting. And what you just said that crossed that over for me is a big part of that is that there's not enough nitrogen in there to support the plant life to then have the food web grow up from it. That's fascinating.
It's probably the primary limiting nutrient in the oceans would be nitrogen. The other one is light. So when we're talking about phytoplankton growing in the world's oceans, they're really restricted to just the surface layer where light penetrates, and below that in the dark ocean, photosynthesis is impossible, and there's much less growth, primary productivity, and biomass being produced.
Yeah, that makes complete sense. That is fascinating. So this could potentially be a total game changer for populating other areas of the ocean that would otherwise not be able to with life. Like I know about nitrogen fixing plants, and I know things like a redbud or a honey locust and how those are able to promote essentially what do they call, the pioneer species, and be able to have whole areas turn back over to, because of the fact they're putting nitrogen back in the soil. So I'd imagine this could potentially be a keystone of some sort. But to not get too ahead of myself, how did this get discovered? I know you mentioned your advisor and
yep. So that's a great question. I think it's a great story. My advisor here, John Zaire, was really one of the pioneers of studying DNA in seawater. And he was kind of moved by this question of nitrogen fixers, looking for them in seawater, and developed a DNA-based assay. It's a PCR type method, which most people are now familiar with. So he developed a way of using PCR to look for nitrogenase. So look for the gene that's encoding this protein that does the business of nitrogen fixation. And so then if you do this PCR assay in seawater, you can then extract all the different versions of nitrogenase from seawater and then sequence those. And so then you get a bunch of DNA sequences that are largely the same, but small variations can be used to distinguish different species, so different organisms that all perform nitrogen fixation will have a slightly different version of that gene. And so he, spent a long time, decades, really surveying the oceans, collecting different versions of this sequence, comparing them, and trying to figure out what organism they would come from. Very little was known about this in the early days, and there weren't a lot of reference sequences. So if you did some sequencing and you got a sequence back, then you compare it to a database and see what does this look like? What is it similar to? And a lot of those sequences would be similar to known nitrogen fixers, typically cyanobacteria. that were known about in seawater, but a lot of them were different enough that we couldn't really tell what they were. And one in particular, which was called Eucinae, for kind of based off of just the DNA sequence. The sequence it looks similar to cyanobacterial sequences, but it wasn't. It couldn't be ascribed to any known organism at the time. And furthermore, it was really abundant. It was found all over the world's oceans. Eventually found at high latitudes, typically where nitrogen fixation wasn't thought to be favorable to organisms who did it. So it was kind of a mystery. It was widespread, abundant, but kind of unknown. Eventually, through some pretty clever techniques, they were able to isolate enough of this organism from seawater to sequence a genome from it. And this is without ever really being able to see it, but they were able to figure out clever ways of concentrating cells to get enough biomass, enough DNA to perform genome sequencing. And when they did that, they developed the genome for this organism and saw that it was missing a lot of very important things. Cyanobacteria are typically photosynthetic organisms that fix carbon using photosynthesis. And this genome had lost that capability. So it was missing part, and really the important parts of photosynthesis, meaning it was unable to fix its own carbon. But I think more interesting than that, it was missing other things which are typically essential in any living organism. It was missing the ability to make its own DNA, to make its own amino acids. It was missing genes that are important for controlling the cell cycle, cell division, and things like many biosynthetic pathways for other compounds were incomplete. So it was kind of, a biosynthetic pathway is like a series of steps that are used to create biological molecules from precursors. And so this genome would show maybe most of the steps are there, but it's missing one or two steps in these pathways, which really renders the whole pathway useless. And it was missing really fundamental things. It can't make its own proteins. And there was really no indication that this genome could survive on its own. It was really a broken genome. And so after that genome was sequenced, I think that was around 2016 or so, it was just assumed that this nitrogen fixer must be living in association with a symbiotic partner that was providing it with the things that it's missing from its genome. And so that was the assumption made based on this this incomplete genome. And that was kind of the understanding of this organism for years, and eventually they did find that indeed it does live in association with an algal host. So it's this small kind of streamlined cyanobacteria which lives inside a larger type of algae. And that was discovered a number of years later by people working in John Zayer's lab, and also by researchers in Japan. They were eventually able to isolate this algae from seawater and determine the conditions that it requires to grow in the lab. And so once that happened, it was a complete game changer for this type of research. For decades, we'd been studying this organism, Eucinae, in the field, in the ocean. And that's very limiting in terms of the types of biology that you can do when you're studying cells, out in the middle of the ocean. It's kind of like looking for a needle in a haystack, and you can't really perform many biological techniques that you can if you have it growing in a culture. And so once this culture was established, it kind of opened the doors for much more kind of advanced types of biological research.
Wow, it's a very Sherlock Holmes adventure. You find a broken genome that you assume, since it can't live on its own, that it has got to it has to have a host, and then it turns out that there is a host. So is this a new species? Is it a new genus in totality? Like, what is this Nitroplast categorized as?
Yeah. It's a new organelle. So it's been demoted. It used to be thought in the days of Eucinae. Eucinae is not really a species name. That just refers to the kind of the DNA sequence. But it was given a kind of a temporary name that sometimes we give to bacteria that before they get an official name. But forget about that name, because that name's out. What our research showed is that it's not its own species. It's actually part of this algae. It's an organelle of this algae, and so therefore it doesn't get its own species name. It's the the algae's name prevails here. So yeah, that is kind of in essence what our latest research has shown, is that it is indeed not a symbiont. It is an organelle, and that is the that's the distinction between two species living together in a symbiotic relationship versus one species with kind of a novel compartment inside of it that is derived from this old symbiotic partner. So at one point they were symbionts living inside an algae, but we've shown now that they've crossed the line from symbiont to organelle.
Okay, so in that case, is this algae now a new iteration of its own species, and that kind of like
yeah.
Okay.
I mean, you could say that. Yeah, the ancestor of this algae did not have a this organelle inside of it, and so it is. Now that it does, is that a new species? Yeah, I guess you might call it that.
How prevalent are they? Like, how I believe at one point you had said that the nitrogen fixing genome was found in a lot of places, but how prevalent is this algae that can produce its own nitrogen?
It's it's very widespread. It's but because its kind of niche is living in these low nitrogen regions of the world's oceans, and maybe that's not exactly true, but it's very widespread. It's very successful, but it doesn't typically reach really high concentrations. So it's not going to be responsible for like an algal bloom that turns the water green. But it is significant because it's so abundant. Even if it's present in low concentrations, it's present over such large regions of the oceans that this biomass adds up. So I mean I'd say this strategy for this alga has turned out to be pretty successful, allowing it this kind of widespread success across the oceans. But it's not something that's gonna grow out of control.
Right. It's not. We're not gonna have to worry about toxic nitrogen fixing algae blooms.
Yeah, I don't think so.
Okay. What implications does this have right now? I guess like part of what I'm asking is also, we're just discovering this now, but has this been out there for decades, hundreds of years? How old is the species, and what implications does having life in these areas that are low nitrogen, low species bearing areas, having more of them in there?
Yeah, I mean, yeah, so it is, I'd say it's a pretty ancient organelle. It's 100 million years old. So these things have been out there for a long time. It's not ancient relative to the chloroplast and mitochondria, which are estimated to be between 1 and 2 billion years old. So it's much more recent if you're comparing it to those organelles. But really, it's been there for a while, and it has been shaping ocean ecosystems. So it's not changing anything, but it is changing our understanding of how these ecosystems work, and it is changing our understanding of like the distribution of nitrogen fixation, where organisms use it. I mean, this is a algae that has pretty much given up the ability to take in nitrogen from its environment, and instead it favors just making its own nitrogen, using the nitroplast and using nitrogenase. And it does this even when there is nitrogen available to it. So it kind of it forces us to rethink our ideas about where and when organisms use nitrogen fixation. It helps us better understand nitrogen cycling in the marine environment. There's, I think, still a lot of questions about how much nitrogen is fixed in the world's oceans, and when and where this occurs, and the organisms that do it.
That's fascinating. It's much older than we may have expected, and it's not necessarily. I realized that my question was inherently skewed, because it was, how is this going to change the world? And it's like, well, actually, they've been here for a while, and it's already impacted everything, which is really a game changer perspective. I used to have another podcast that was actually called the Nitrogen Podcast, and the whole point of that was, nitrogen is the most abundant, in our atmosphere, but it's something we breathe in, and we need it, but we don't think about it because it's everywhere. And this kind of is very much of that kin. It turns out something we didn't think was out there is actually very prevalent, and it's been around for a while.
That is, that's a very common theme in the field of marine science. I mean, there's a lot of unknowns, and there's a lot of things out there that we're surprised to find.
Yeah, definitely.
I mean, I think just maybe a month or so after we published our paper, there's another significant paper published in Nature, which describes another nitrogen fixing symbiosis that had gone unnoticed for a long time. They don't have the data to show if it's a nitroplast or not. I think it probably is, but it's just another example of this kind of untold diversity of marine microbes that are very difficult to study.
Yeah, that's fascinating. So it's more prevalent even outside of this one instance, but it actually might be out there in several more instances.
Yeah, I would not be surprised.
Wow. That's a really stunning example of nature finding a way.
Yeah.
Well, thank you so much for your time. Was there anything else before we pause the recording?
I mean, I could talk for for however long you want. I think, I think you did a good job of hitting on the important points in this research, and so I'm happy to leave it at that.
Well, thank you again. I appreciate your time, and I would love to have you on again. So I'll definitely pause the recording, and if there's anything else that comes up, definitely let me know.
Okay. Well, yeah. Thank you.